Okay, great, thank you. Thank you very much for the invitation to speak with all of you. My name is Jeff Bowman. I'm an assistant professor at Scripps Institution of Oceanography. The work that I do is quite far from nutrition and agricultural studies, but there's a lot of commonalities across these different microbial ecosystems. So I look forward to talking with you all today about what I do. So there's going to be a couple different parts to this talk. And first of all, if I move the pointer over the screen, can everybody see this?
Yes, yes, we can see your screen.
Okay, thank you. So I'll give a little bit of an introduction, and I'll describe some of the methods, the PAPRICA technique that we use for metabolic inference. I'll then go into a practical tutorial on how to actually use PAPRICA to perform an analysis, and then I'll end with a little bit of a demonstration of some different applications for PAPRICA. So as I mentioned before, I am primarily an oceanographer. I primarily work in high latitude systems, and I developed a tool, PAPRICA, as a tool for trying to estimate the metabolic potential of different communities that we were investigating. We have very few metagenomes available to us in these different environments, very difficult to get samples, and so we needed some alternate techniques. Nonetheless, a lot of the organisms that we studied are analogous to organisms that might be present in different microbial ecosystems, and some examples of these are shown here. So here we have some microbial eukaryotes, and here we have bacteria and archaea. Great, thank you. Okay, so what I'm going to start off with is defining the problem that we're trying to solve here. So in this case, we have 16S reads, a library of 16S reads, describing the structure of some community. And we want to perform a metabolic inference on that community so that we can understand the functional potential that is contained within that community. So why might we want to conduct a metabolic inference? Sequencing has gotten very cheap, but it is still impractical to sequence metagenomes for very large sample sets. We would not, for example, want to try to sequence metagenomes for many hundreds or many thousands of samples, but these are the size of the data sets that we often need to collect in order to apply the statistics that we want to do. Also, even very deep metagenomes still miss rare genes. So even if you spend a lot of money to sequence a given sample to a very extreme depth, there's no guarantee that you're going to capture all of the genes, examples of all the genes that are present. A metabolic inference can help you fill in some of those gaps. And finally, it's useful to have a model of the distribution of metabolic functions as we currently know them. So this is much more common in other fields. This is less common in microbial ecology, but here what we've really done is create a model of what we think should be present functionally based on what is present taxonomically. And interesting deviations between what we actually see and what the model predicts can be useful in its own way, and I'll talk a little bit more about that later. So PAPRICA is certainly not the only piece of software out there to help you do this type of work, so it's worth mentioning a couple of others. One is PyCrust, which is probably the most widely used of the available methods, and another is Tax4Fun, which is an R-based package. One of the major differences between PAPRICA and PyCrust and Tax4Fun is that PAPRICA does not rely on a closed OTU description of community structure. It's actually based on the phylogenetic description of community structure. And in addition to that, it provides a start-to-finish solution for both community structure analysis and metabolic inference. And I'll talk more about that shortly. Okay, so there are really three different parts to PAPRICA. One is the construction of the database, and for most users, this is already done in advance for you. Only an advanced user would need to go in and tweak the database and build their own. A second is the analysis part. This is where you take your 16S sequences and insert them into the pipeline. And then the third part is confidence scoring, which gives you some estimate of how accurate your predictions are. Well, I'm going to talk about each of these three categories in a certain amount of detail, but before I start on that, I want to offer the following caveats.
Any of the metabolic inference methods that are out there are only as good as our available genome annotations, which we know are not necessarily always accurate or complete. It's also dependent on the diversity of completed genomes that we have available to us, and we know that there are certainly gaps, taxonomic gaps in the diversity of completed genomes available. And finally, our knowledge of enzymes and metabolic pathways. These are also somewhat weak. We don't always know what all enzymes are doing. We don't know all the metabolic pathways that are out there and the different functions that they might be associated with in different organisms. A final point of limitation is going to be genomic plasticity. And this is the loss or acquisition of new genetic material into the genome. There's obviously deviations between how this works and the the vertical taxonomic structure. And I'll talk more about genomic plasticity later. Okay, so first I'm going to start off by describing the construction of the database. Now, the first thing that we do is we obtain all of the completed genomes from GenBank. So, for example, during the last build of the PAPRICA database, there were about 6,000 bacterial and 2,000 archaeal genomes available to us. For the eukaryotes, we rely on the eukaryotic transcriptomes in the Marine Microbial Eukaryotic Transcriptome Sequencing Project, or MMETSP. And there's about 649 of those available. We look at those genomes and we extract the 16S rRNA genes from those different genomes, and then we use them to build a reference tree. This is a graphical depiction of what that reference tree looks like. We then predict the metabolic pathways for each of the genomes that we're using to build the database. And then for each point on that reference tree, we find a consensus set of metabolic pathways and enzymes. And we also collect this information that is shown over here on the left side of the screen. So we have the metabolic pathways and the enzymes. We also look at the GC content, 16S genes, genome length, etc., for each internal and terminal node on that tree. This is a picture of what that would actually look like in practice. So if we zoom in on one little portion of that tree, this would be the Phylogenetibacter genus. This is a very important marine bacterium that gets a lot of attention, but it only has 3 completed genomes available to describe it. Notice that on this little subset of the tree, there are these 3 terminal nodes given by the completed genomes, and then there are these 2 internal nodes. So we know what the genetic content is out here at the terminal nodes on the tree. For these 2 internal nodes, we simply use the consensus. We use all of the enzymes and pathways that are shared. For example, here, it would be shared by all 3 genomes. For this point, by just these 2 genomes. Once the database is constructed, we can go ahead and do our analysis. So for our analysis, we start with a library of 16S sequences. We align the query reads to the reference sequences, and then we place them on the same tree that we use in the database. So this, again, is that tree. You'll notice that some of the edges on this tree now are fattened. They're fattened proportional to the number of query reads that have been placed to them. So this is really a graphical depiction of the structure of our community. Now, this is very different than how most people do community structure, so it's worth a little bit of further discussion. One thing that we've done is to try to understand whether within sample or between sample distance is well retained by this method, which indicates that it's working well. So what you see here on the left side of this plot is the distance between a set of samples given by the method used by PAPRICA and distance between a set of samples given by Silva OTUs at 97% similarity. And the data set that's being used here is are the the 16S sequences from the Terra Marine expedition, which I'll talk about a little bit more later. The main point here is that there's really good fidelity between the distances given by PAPRICA and the distances given by Silva. You'll notice that the distances are higher for Silva. This is the 1-to-1 line here, but nonetheless, where distance is high here, distance is high here. So once we've placed all those reads, we can simply extract the genomic information for each placement. And we have genomic information both for the internal nodes on the tree as well as those terminal nodes. The final thing that we need to do is generate a confidence score for these samples. So the confidence scoring is a little bit more complicated. But what we do here is first off evaluate the relative size of the core genome. So I'm going to show this figure again. Here we have a hypothetical internal node, and we have two terminal nodes. The core genome size the core genome is the genetic information that is shared between these 2 points. The other part of the genome is the accessory genome. Now, the relative size of the core genome relative to the overall genome gives you a sense of how likely it is that genetic information will appear at this internal node. So we take that information. We also use an indicator of genomic plasticity for all of the clade members. Now, the way that we do this is with a parameter that we call phi, which we originally created to try to measure horizontal gene transfer between different genomes. And here we're simply applying it to understanding the stability of the different genomes in our database. So the basic way that the phi parameter works is imagine that you have a single bacterium that splits into 2 daughter cells, and those 2 daughter cells and their lineages start to diverge genetically with respect to time. So here we have divergence, here we have time. Now, the basic idea here is that if we have a conserved gene, such as the 16S rRNA gene, it's going to diverge at one rate. The genome, the functional aspects of the genome without horizontal gene transfer will diverge at a faster rate. And as we start to add in horizontal gene transfer events or genome streamlining events, we will get increases in the amount of divergence. So the plasticity is some function of these 2 measures of distance between the conserved gene and the genome itself. So we figure the genome distance from compositional vectors. It's simply the distance between predicted proteomes via compositional vectors. And we figure the distance between the 16S rRNA gene using the standard methods. So if you plot those 2 distances for all the genomes in our analysis, this is the type of relationship that you get. So what we look for is genome to genome comparisons that fall, what we look for is genomes that fall far from this line of best fit. Genomes that fall to this side of it have relatively low plasticity compared to the bulk population of genomes, and genomes that fall to this side of that line have high genomic plasticity. If we take the residuals for each of those genomes and we do we sum them up all the way across so that each genome is being compared to all other genomes, we can get an indication of the relative plasticity of all of the genomes. So this is our phi parameter here. This is our indicator of relative plasticity. These are each of the genomes that went into the analysis. This is a somewhat older plot, so there are far fewer genomes than are currently present within the PAPRICA database. But what you'll notice here is that organisms that have a high phi parameter, or bacteria and archaea that have a high phi parameter, are consistent with clades of bacteria that are known to have high rates of horizontal gene transfer or extremely streamlined genomes. All right, so we have phi, our measured genomic plasticity. We have our mean clade genome size, and we have our mean core genome size. We use this information to build a confidence calculation for each node. Taking the mean of all of the nodes for a given sample gives us a confidence score for that sample. All right, so that's really how PAPRICA works from a database and analysis standpoint. Now, there are 3 ways to actually implement PAPRICA to actually run it. The preferred way is locally on a workstation or a server. You can do this on OS X or Linux, and if you're a Windows user, you can compile the various component pieces of software with Sigwin. Additionally, we also provide an Amazon machine instance. So if you're an Amazon Elastic Cloud user, you can simply find the machine instance online and use it as is. And last, we provide a virtual box. And this is really for testing, for people that just want to get an environment that they know the software works in, to see if it will help them with their analysis. You can download the virtual box. All of the dependencies are already installed, and PAPRICA is already up and running. We don't recommend this for production work because there's significant overhead to using the virtual box appliance. But nonetheless, it's an easy way to see if PAPRICA is going to work for you. All right. When you actually get around to running PAPRICA, there are a couple things that you need to know. There are two shell scripts that really execute, that really do the database building or execute the analysis. So these shell scripts themselves run a different set of Python scripts where the actual commands are executed. The PAPRICA build script is not necessarily for most users. This is only if you want to build the database yourself. Pre-built databases are provided. For most users, you're only going to have to interact with this PAPRICA run script. And the way that you execute it is by typing PAPRICA run and then the input FASTA file that you want to analyze and the domain that you want to analyze it against, either archaea, bacteria, or eukarya. And this script does the actual analysis. It executes a series of Python scripts given a FASTA file that has the level of quality control applied to it that you want. And this is the link to the GitHub site where all of these scripts and the pre-built database are located. All right. So this is what the PAPRICA run script actually looks like if you open it up. So you can see that some variables are being set here. You don't need to do anything with this. And then these two Python scripts, where the bulk of the work is actually being done, are automatically executed for you. Now, there are a couple of things that you might want to change within this. So one thing that we've done is we've built some parallelization into this. The infernal aligner is inherently parallelized. It will automatically make use of the available cores in your computer. However, pplacer, which we use for phylogenetic placement, does not. So this flag here, this splits flag, can be changed to a number higher than 1 to automatically split your query FASTA file apart and automatically put it back together at the end. This greatly speeds up your analysis. You can also set flags here.
Wait, sorry, Jeff. Sorry, Jeff.
Libraries, if you know that you want to say subsample to the size of the smallest library, you can set that here as well.
Sorry, Jeff.
What I had planned to do at this point in time was to run a little video that just shows how you can execute PAPRICA on two different FASTA files here. So here we've moved the PAPRICA run script into our working directory. We're opening it up to make some modifications to this first line. We're going to increase the number of splits here so that it will split that input file apart. We close this out now. And then we can go ahead and run the, execute the script to run the analysis. So here we're running on this file, summer.fasta, where we are running it against the bacteria database. And now we can see that this is the output from the standard CM align command. It's going to go through and very efficiently align the query reads to the reference alignment. And now we've started to move into the phylogenetic placement step for some of the subset files or some of the split files. I'm not going to let that run to completion because it will take a few minutes, and I know that we're running a little bit short on time, but this just simply illustrates, you can see here pplacer running, actually a total of 48 processes, I believe, in response to us splitting that input file apart. PAPRICA will automatically join all of those together at the end so that you have one single output file. We'll go ahead and stop this. Oops. And once that analysis is complete, these are the files. And once that analysis is complete, these are the files that would have been created for us. So this was our input file here, summer.fasta, and these are all of the output files that have been created by PAPRICA. So we're going to look at a couple of the key ones now. If we open this file, summer bacteria pathways.csv, this is the pathways by edge. So each of these numbers up here is one edge in our phylogenetic tree. This is a metabolic pathway, and we can see the number of pathways that are attributed to that edge within this sample. So a lot of these are 0, but here we can see numbers coming up. Now you'll notice that these are not integers, which might be surprising, but one advantage to using PAPRICA is that all of the information you get back has been normalized for multiple 16S RNA genes. So we know how many 16S genes are present in each of those edges on that reference tree. So we simply divide the number of pathways that we estimate to be there by the number of 16S genes present in each of those genomes to get an accurate number of metabolic pathways present within the sample. Sum are bacteria sum pathways.csv is simply summing across all of the columns in that previous files. This is the actual metabolic structure in pathway space for this particular sample. And finally, summary bacteria edgedata.csv. This is looks a little bit messy, but if you opened it in Excel as a spreadsheet, all the columns would be aligned. It would look rather clean. So what we have here in the first column is the edge number. This is the number of times that edge has appeared. We have the posterior probability of placement, some other useful information about the quality of the placement on that phylogenetic tree. We have a taxon name, the number of 16S genes present within that genome, a corrected number of edges, and other information that will help you make some ecological inferences about what your community is doing. All right, so I'm running very short on time, so I'm not going to spend a lot of time going into the applications. But there are a couple of things that you can do with PAPRICA, in addition to just exploring the metabolic content in your samples. One of the original things that we did was use it to look for functional redundancy in microbial communities. I'm going to go through this very quickly, but just to illustrate that if you have a set of 16S sequences and you convert those sequences into an estimate of the metabolic structure, you can do things like this. You can look at the number of different phylogenetic edges that a given pathway has occurred in. Pathways, for example, that are relatively abundant in your sample but appear in very few edges, you might consider to have low functional redundancy. And so if that edge or that bacterium disappeared from your sample, you would have some significant loss of functions. That's what we're exploring here. It's also useful for identifying metabolic functions that you simply don't anticipate being present within your sample. And a nice way to think about this is that PAPRICA and the other techniques that do metabolic inference really just formalize what we already do already. If you, for example, obtain an isolate from the environment and you sequence its 16S gene, you're going to get the taxonomy of that gene and you're going to go flipping through the literature to try to understand what kinds of things that bacterium is likely to do. PAPRICA really just speeds up and formalizes that process. It's looking across all of the related genomes and trying to make a reasonable estimate of what that new strain might do. So one really great example of this is, so I work a lot on sea ice and sea ice microbial communities. There was a sea ice bacterium here belonging to the Corallia margarita genus that nobody really understood what it might be doing within sea ice. It has all of the genes for doing nitrogen fixation, but the very short paper that described this reference genome from a temperate environment made no mention of its nitrogen fixing capabilities. Therefore, we were all sort of missing that it might be playing this ecological function within sea ice until we ran the PAPRICA analysis, and these functions immediately popped out. And finally, and I alluded to this a little bit before, it's very useful for identifying what we might call microbial dark matter, or places where we have a poor understanding of the functional content of microbial genomes. And so this is using the Terra Oceans sample set. This is a global metagenomic survey of the oceans. And what we've done here is we've looked at Bray-Curtis distance between predicted metagenomes up against observed metagenomes. Places where there are strong deviations here between these two indicate that we have a weak understanding of the genetic contents of the microbes that are present. And you can play with this a little bit to start getting a sense of, well, where in the ocean do we have a poor understanding of the genetic contents of the resident bacteria? And that's what we've tried to do here. All right, so I'm going to end very quickly here, but one final thing that I will mention is that we've extended PAPRICA to do both metagenomic and metatranscriptomic analysis. And the reason that we've done this is that we've gone through this effort of organizing this database phylogenetically. And so we've got all of these reference genomes that are organized phylogenetically, and we know quite a bit about the content of those genomes. This is a perfect setup for mapping either metagenomic or metatranscriptomic reads back into that space. And so we've spent quite a bit of time building modules to do that, and I would encourage you to, to experiment with those and see how they're helpful for you. So in the interest of time, I'm not going to go through this in any great detail, but I would encourage anybody who is interested in this type of analysis to go ahead and contact me, and I'd be happy to talk to you more about it. So I think I'm at the end of my time here, so I'd like to
we have some space.
For discussion. Go ahead.
We have some space time-wise if you want to go a little bit deeper now. It's possible.
Okay, great. So I'll do that. It will just take an extra minute or two. So what we do here is we build a database by collapsing all of the non-redundant coding sequences into a very large CSV file that has everything indexed by genomes, the CDS, the sequence, and then we can use, depending on your analysis, whether you're doing metagenomics or metatranscriptomics, we can use either the diamond search method or BWA to search against the PAPRICA database. And what we get back from that is, in the example of metatranscriptomes, we get the number of reads that have mapped to a given coding sequence within a given genome, and it allows you to break your metatranscriptomes apart into a series of individual transcriptomes, if that makes sense. So you're really looking at a per genome shift in gene expression, which can be very important to trying to understand the true pattern of upregulation or downregulation in your experiment. So this is an example of that. We're working with a fairly large metatranscriptome data set from the West Antarctic Peninsula marine ecosystem right now. What you're looking at here, this is just genome abundance based on the number of reads that have mapped to different genomes. These numbers here are all indicating different marine phytoplankton that were sequenced within the marine microbial eukaryotic transcriptome sequencing project. So this is where the eukaryotic genomes are coming from in PAPRICA. These are different time points across this particular experiment, and the color here is giving you the number of, or the abundance of that genome. So we can see that this genome here was very abundant, and it's kind of decreasing across the experiment. Here, this genome is very low in abundance, and becomes abundant later in the experiment. Now what we try to do is, for each of these genomes, we pull out specific points of comparison, and so we can look at each genome individually in isolation. Therefore, even if this genome is decreasing in abundance across the time, we can still look at genes that are being upregulated within it, and we can still capture that signal. If we didn't do it on a genome by genome basis, we would lose this. So this is your standard DEC2 style plot. So we have log fold change on the y-axis and mean normalized counts on the x-axis. So these are the more abundant transcripts. These are the ones that are changing more. And red here indicates a significant change between these 2 points of comparison. So this has been looking across all possible transition points in the time series that defines this experiment for this genome. And these are the different enzymes that are significantly differentially expressed. Red indicates upregulation, blue indicates downregulation. And so now we can start to pull apart the ecological story that's underlying these this differential expression. And these are enzyme condition numbers for the different coding sequences that we are evaluating. Okay, so that's all that I have. I would point you to a couple of different sites here. We have an extensive set of tutorials that you can find on my website here. And this is always evolving. We've actually added some new features to PAPRICA that I didn't get to talk about today, and they will be reflected very soon within the tutorials. The code itself, of course, is at our GitHub account, including the pre-built databases, and our VirtualBox appliance can be found here. And I wanted to kind of end with this figure. This is simply a graphical depiction of the metabolic pathways contained within the PAPRICA database. So here we have all of our genomes. So each of these rows is a genome for the domain bacteria. Each of the columns is a pathway. And so we can get a sense of this really nice taxonomic distribution of the different metabolic pathways across all these genomes, which in this plot have been organized phylogenetically. So thank you very much for your time and the opportunity to speak with you, and I'd be happy to take any questions.
Okay, thank you.